Hello, everyone. I hope everyone had a wonderful weekend and we're ready to tear into a few more chapters of Dr. Rudy's Neurobiology of Learning and Memory textbook this week. But before we get going, I have a couple of housekeeping things to talk about. And, well, a couple, meaning two, means I have two things to talk about. Number one is the syllabus, and number two is the exam I just returned to you on the weekend. So I've made a small adjustment to the syllabus as I actually noticed an error on it. I actually had scheduled this week starting on Sunday rather than Monday. So I had it starting on the 26th rather than the 27th. So I adjusted that, and I figure that we will now cover up until and including chapter 12 this week, not chapter 14. And so that means the exam will cover chapters 5 through 12. Now, I posted the revised syllabus on the website, so you have it for your information. Okay, so that's the syllabus. And I have a few notes about the exam. First of all, and most importantly, I want to acknowledge that most of the class did extremely well in the exam. So the next one will be twice as hard. No, just kidding. Yeah. But really, more than half the class got A's, and the class average was 91%. And that's exactly what I expect from a class full of serious students who are, for the most part, planning on careers in science or medical studies. Anyway, well done. That was a superb showing on this first exam. However, if you did not do as well as you expected, you absolutely need to come to office hours. And when you do so, come armed with questions. Review the answers I posted on the website and identify issues that you still might not quite grasp. That's why I post the answers, so they can help you. Now, keep in mind that this material keeps building. If you want to be able to keep up through the rest of the course, you will need the foundation that we built in the first 2 weeks. I mean, we're going to be covering some more circuits and systems levels of analyses, but we will continue to come back to the underlying principles that put those circuits and systems together. For example, you will only be able to keep up in the chapters where we examine consolidation of memory and forgetting if you have a sound comprehension of the actions of kinases and phosphatases in structuring the synapse in the first place. So please take advantage of the opportunity that I am offering. I have office hours every Tuesday and Thursday, and I'm happy to say that not a single one has passed that doesn't have students in attendance asking questions, reviewing the materials with me, chatting about science. So come and join us. It's kind of fun. Okay. And then there were a few issues that came up in the test, and a few is 3. So I don't want to belabor these points, but they are important. One point is about copying and pasting answers. The second is submitting exams on time, and the third is following the instructions. So bear with me for just a minute while I go through this. Let's start with copying and pasting. It's very clear that some students think that a proper way to answer an essay question is to copy something that's published, paste it into your document, and then change the words. Now, this is actually the definition of plagiarism. Even if you change every word, you've taken somebody else's work, their thoughts, their structure of the written material, and represented it as your own. And the fact that you changed many or most of the words demonstrates that you know that what you are doing is wrong. You know that it's the worst form of cheating. And I'm going to warn you that the consequences are severe. The question on the exam for which this was most apparent was the first short essay question about the Morris water maze. 11 students wrote their answers by copying the same text that was available online and changing some words. Now, if you change the words, and so do 10 other students, don't you think that I can see the same sentences and the same paragraph structures coming up in the writing of all 11 students? And don't you realize that if you can find that source paper online, so can everyone else? Okay, so I let it slide this time because I wanted to formally address it with everyone rather than dealing with all the ramifications, because they get very ugly. However, next time, I will submit every instance of plagiarism to Turnitin.com, and all plagiarized text will be identified, and I'll be forced to prosecute it through the official channels of the university. So please do not force me to do this. All writing must be independent and original. Do not copy and paste any text from anywhere, and do not work with any other student in formulating your answers. Okay, and to the 44 students who did not copy and paste, I apologize for perhaps sounding like I'm scolding everyone. I don't mean that. I appreciate the good work of the large majority of the students in my course, so keep it up. I'm also going to say a few words about late exams. I told students that there was a 10-point penalty per day for late exams, and nevertheless 5 exams were submitted late. And one was actually not finalized until Saturday because of an error in the submission. Now, admittedly, I was delayed in grading them because I have a lot of other responsibilities to the university, which are being complicated by the pandemic situation. So it's been a tough week. Hopefully better this coming week and beyond. But here's an issue. I like to post the answers to the exams as early as I can so everyone can take advantage of checking their answers. I add all kinds of annotations about which answers are correct and so on. And I hope that that's useful material, but I couldn't do that this week. So I just put them up on the weekend after I got all the exams in. So I'm adjusting the policy for the next test in order to make certain that it's fair for all students who do turn in their tests on time, which is the vast majority, of course. So for test number 2, there will again be a 10-point penalty per day for late exams, but any exams not submitted by 9 o'clock on Wednesday morning will not be accepted, because I'm going to post the answers on Wednesday morning at the latest. And of course, I can't accept any late exam after the answers are posted. And then for test number 3, and I already talked about this before, I can't accept late exams at all because it's the end of the course. All right. Okay, and my third and last point about exams is following instructions. And please pay attention to this. Don't alter the form. Just fill in your exam form in the spaces that I allocated for the answers. Don't delete the questions and make up your own version, because all of your bulleting and formatting makes it very difficult for me to give you any kind of meaningful comments. And do not convert your exam to PDF. I'm going to restrict the submissions on the next one, so PDFs will not be accepted by the website, because I can't then provide comments on your exam paper about, what part of your answer is correct or what you've missed or whatever. And I do like to provide those comments to students because, again, I hope they're helpful. And on that note, I hope everybody was able to see my comments on their answers and that they were actually helpful. Okay, and the last part of the point about following instructions is please read the questions carefully. Some questions have multiple parts. For example, question number 26 was, describe the mechanisms of presynaptic plasticity during associative conditioning in aplesia. Include the experimental paradigm with a CS+, a CS-, and a US. What changes happen to signaling mechanisms in the sensory axon terminal? What signals encode the CS plus US contiguity? And how are these changes affected by postsynaptic activity? So you can see that that question has multiple parts to it. And many students who are writing otherwise brilliant answers missed one or more parts of the question, like not describing how the postsynaptic activity affects the presynaptic changes. So in this case, even though an answer that might otherwise have been just excellent and got a perfect grade, I couldn't reward full points. So please be careful to read and answer each question fully on the upcoming exams. Okay, so I hate to go on and on about that, but I'm glad to have all of that cleared up. If anybody has any questions, please do not hesitate to contact me. And again, I want to emphasize that the class, for the most part, did great. So pat yourselves on the back. And if you didn't do great, come to office hours, or just come to office hours anyway. I'd love to talk with everybody about science and all, okay? All right, so let's start chapter 9. This is actually a really short chapter because Dr. Rudy decided when he wrote the textbook that he would describe behavioral tests in chapter 9. But I moved that information to chapter 1 because I think it's important that we all understand the behavioral paradigms before we start to talk about the way that they've been used in the various experiments that we discussed in chapters 1 through 8. So I invite you to read Dr. Rudy's descriptions of the behavioral tests anyway, so it will reinforce some of the things I already told you in chapter 1. So in this chapter 9, I'm going to focus on some related issues, assuming that we're already well acquainted with the behavioral tests. I'm going to talk to you briefly today about LTP and behavior. What a surprise. It's like we've never done that before, right? But this time, I'm going to focus upon some of the experimental manipulations that are conducted with animals to see how the environment or other kinds of perhaps more intrusive interventions can impact learning and memory. So rather than talking about behavioral tests of learning and memory, I'm going to focus on the actual manipulations that are performed before the animals are put into those tests. I'm going to focus on enriched and impoverished environments and pharmacological manipulations or genetic manipulations and so on. And well, since this chapter is so short, I'm going to try to get chapter 10 posted up soon also, so you can continue to move forward. So before I start to talk about all of the behavioral kinds of manipulations and drug manipulations and so on that we do with living animals, and in keeping with the content of Dr. Rudy's textbook, I want to start with a reminder that LTP or LTD is simply an experimental manipulation. Now we think of it as a model of learning, but it's clearly not learning itself. In fact, a few students have asked me questions about this, so I think it's appropriate to take a look again. Your textbook, chapter 9, starts with a few paragraphs explaining that LTP is an artificial preparation. So let's talk about that. Of course, the hippocampal section is a nice mini circuit consisting of 3 synapses. However, it doesn't represent all the diffuse distributed synapses that are thought to underlie the storage of any particular memory code. And as we move into talking more about circuits and systems, you're going to see more and more how this trisynaptic mini circuit really doesn't fulfill all of the requirements of learning. It's just a section of tissue floating in a little bath with electrodes in it. Nevertheless, it has helped to reveal a variety of the synaptic mechanisms that we've already discussed and is broadly used to understand errors that happen in neurodevelopmental disorders or traumatic brain injury or age-associated cognitive impairment, and so on. The setup for in vitro LTP is shown on the right side of this slide. Now, I know that's a very busy looking kind of picture, but I want you to see some of the things that actually go on when we do electrophysiology in a hippocampal section. You should be able to see a polycarbonate chamber here with a well in it, submerged in a cold chemical bath with roughly physiological concentrations of ions and glucose, and with oxygen bubbling through the bath to try to keep the tissue as viable as possible. That viability is generally limited to about 6 to 8 hours, and recordings beyond that time are typically compromised. So you could argue that the tissue is compromised from the outset anyway, as it's being cut out of the brain, slashing through all the neuronal connections that connect it together with the rest of the brain, and now it's sitting in an ice-cold bath where chemical reactions are actually compromised because the kinetics of those reactions are slowed down by being in the cold bath. Your body temperature is kept a lot warmer because those chemical interactions function better in a warm environment. So clearly, an animal is not learning anything in a slice preparation. It's a piece of tissue, and that is all that is so compromised. But it has told us nearly everything we know today about the cellular biology of learning and memory. An alternative is to use in vivo electrophysiology. An advantage of this approach is that the consequences of potentiation or depotentiation can be assessed in the living animal. But it's also a much trickier surgical manipulation than the straightforward slice preparation that we saw in the previous slide. And, well, at least the experience of my lab is that the extra hippocampal brain regions you might like to target in a setup such as this might not be so amenable to LTP-like manipulations. Remember that the hippocampal slice contains fields of dendritic arbors, where you can kind of jab in an electrode and get extracellular recordings that represent the average function of many aligned and virtually identical granule or pyramidal cells. So in fact, the photograph on this slide is taken from my lab when one of my graduate students performed a series of experimental manipulations examining the impacts of tetanic stimulation applied to the bed nucleus of stria terminalis, or BNST. This is a part of the limbic system. Now, she had recording electrodes in the paraventricular nucleus of the hypothalamus, or PVN. And this is the linchpin of signaling in the hypothalamic pituitary adrenal, or HPA axis. This is responsible for regulation of cortisol in response to stressful situations. So we examined changes in behavioral indices of anxiety and depression-related behaviors and alterations in hormonal responses after potentiating stimulation of the BNST, of this limbic structure. Now, the outcomes were very complex, and I'm going to avoid spending the next hour describing those outcomes. But it's sufficient to say that although we did publish some important data from these manipulations, the BNST is a highly complex structure that contains multiple inhibitory and excitatory circuits. So it's definitely not as easy to manipulate and interpret as the standard hippocampal sections are. Now, in the photograph, you should be able to see an anesthetized rat with microelectrodes surgically implanted into the BNST and, well, in the PVN. using a stereotaxic apparatus. Now, all of this is contained inside a Faraday cage to electronically isolate the preparation, and the electrodes are connected to electronic hardware and a computer with specialized software to control the stimulation and to monitor the field potential responses. Nevertheless, I want to remind you again that even in an in vivo preparation, nothing is learned here. I can alter synaptic strength and easily disrupt the encoding of some previously learned event or association, but it's not like a few pulses of tetanic stimulation confers some new declarative memory or skill. We're really hammering the synapses in the field of the stimulation. Yet, once again, it's the best model we have, and it continues to reveal new information about the neurobiology of learning and memory every day. So in fact, if we want to study actual learning and memory, whether we're using animal models or humans, we need to be very careful about how we interpret our manipulations. We measure change in behavior, but there are many variables that can contribute to the change in behavior, and they may not all represent learning. So let's just say that we're going to evaluate the impact of some manipulation on learning in the Morris water maze, or the novel object recognition test, or fear conditioning, or whatever behavioral test of learning and memory you prefer. We find that the animals that experienced the manipulation are either impaired at the learning outcome, or their performance is enhanced relative to the appropriate controls. Should we automatically conclude that they learned better? Yeah, it's tempting to take this kind of superficial perspective, and we see it in published papers quite often. And when you're running a research lab and you get an exciting outcome, it's easy to jump to this kind of conclusion. But there may be other variables that may impact the performance. So the effect of your manipulation may not be a direct effect on learning. So the first thing we need to consider is the experience of the subject at the time of the training or testing. If, for example, sensory function is altered, it could impact performance without impacting the learning apparatus in any way. If your manipulation altered the attentional ability of the subjects, this could also compromise the performance. Or if your manipulation altered the higher level cognitive perception of the stimuli, you could impair performance. But none of these impacts would actually have any direct effect upon the actual learning apparatus then. Similarly, your manipulation could affect motivation. If you disrupt, or for that matter enhance, the animal's motivation to engage in the behavioral task, or increase the salience of the reward that is offered in the experimental manipulation, this could alter performance, but not learning capacity. For example, if you administered an anxiogenic drug like FG7142, a drug that increases anxiety in humans, you might increase the animal's motivation to find the safe chamber in the Barnes maze, or you might disrupt the connection between freezing and fear conditioning, as the animal's freezing whether the cue is present or absent. And of course, in the opposite case, diazepam or Valium may decrease the motivation to find the escape in the Barnes maze, or diminish freezing in the fear conditioning test. So hopefully you can see that all you've done is alter the animal's subjective motivation and not actually altered the learning itself. So you might also impact the animal's emotional well-being making the animal hyper alert and responsive, for example. So you may not be measuring what you think you are in, let's say, a fear potentiated startle. And of course, there's the locomotor problem. Many drugs have stimulant or sedative properties. So if you were to administer them and not account for those variables, you may also end up with a misinterpretation where you think the animal has an altered learning capacity, and really all it has is a locomotor excess or impairment that's interfering with the performance on the test of learning and memory. So overall, deficits in subjective experience, motivation, emotion, and/or motor function could affect outcomes without directly impacting learning itself. The effect on learning is only a secondary consequence, and any conclusion that the learning apparatus or capability was altered would be incorrect. This is actually known as the learning performance distinction. And we have to be extremely careful in our interpretations of our various tests of learning and memory so that we don't mistakenly interpret impacts upon the learning and memory apparatus when really all we've done is impact one or more of these variables. So with all those caveats in mind, those important considerations, let's look at some types of manipulations that are commonly used in studies of the neurobiology of learning and memory in animals. Once again, I'm not going to be focusing here on the specific tests that we've already considered. I'm going to talk about environmental manipulations and pharmacological challenges and genetic manipulations. So I'm going to start with the environmental manipulations, including enriched or impoverished environments and manipulations of stress. All the photographs on this page also come from my lab. The photographs on the right depict our complex, enriched, and austere, impoverished housing conditions. In the top one, you can see what we call an enriched environment. Of course, it's actually quite impoverished compared to the fields and sewers in which wild rats live. Those are truly enriched environments for rats. Our enriched environments even pale in comparison probably to the circumstances Hebb used when he brought his rats home to play with his children. But our enrichment does provide multiple levels in the housing cage with bedding to burrow in, shelters to huddle in, food to forage and protect, and so many other opportunities for species-specific behaviors. And of course, we house our rats in social groups in these environments. And then in the lower picture, you can see our impoverished environments, in which we attempt to minimize the opportunities for species-specific behaviors and social interactions. Most studies of the neurobiology of learning and memory, including Donald Hebb's, have focused upon the beneficial impacts of enriched environments, and few have actually examined the impact of impoverishment. But again, these are relative terms, as even Donald Hebb's house was not as enriching as a sewer if you happen to be a rat that I guess doesn't mind that kind of thing. But we have used our enriched and impoverished environments mostly to study vulnerability for pathological behaviors that are commonly present in autism and other neurodevelopmental disorders. And we haven't yet used them very much in relation to the neurobiology of learning and memory, an area in which we might get more deeply involved. The other photograph depicts one of our studies on the impacts of stress. I'm sure you're all aware that stress can impact learning. We're all stressed, right? I'm stressed, you're stressed, everybody's stressed. But if you're overly stressed before an exam,